Every function in your body depends on chemistry and on electricity. Membrane potentials, ion gradients, nerve signaling. These bioelectrical processes coordinate how our cells work. Modern medicine targets biochemical pathways with remarkable precision, but the electrical dimension is often left unaddressed.
Start with the whole body. Every organ in it, brain, heart, muscles, skin, behaves electrically, and that behavior begins one level down in the single cell. Physiology runs on two regulatory systems at once: chemical regulation, hormones, neurotransmitters, metabolites, and the medicines that act on them, and electrical regulation, membrane potentials, ionic gradients, excitability, and bioelectric signaling between cells. They are not separable. Every chemical event in the body has an electrical consequence, and every electrical event has a chemical one. None of this is new. In 1791, Galvani showed that a nerve could command a muscle. Volta, arguing with him, built the battery. Du Bois-Reymond measured the current that flows from an injury. Hodgkin and Huxley gave the nerve impulse its exact mathematics and a Nobel Prize. The patch clamp, the atomic structure of the potassium channel, and the mechanosensitive piezo channels each followed one continuous line of physiology. There are roughly 37 trillion cells in that body, and each one holds a voltage across its outer membrane. That voltage is stored as ion gradients. Sodium concentrated outside the cell, potassium inside, calcium held very low in the cytoplasm, chloride largely outside. Charge separated across a very thin barrier. The separation is actively maintained by the sodium-potassium ATPase, which spends ATP to move 3 sodium ions out for every 2 potassium ions in. Holding that gradient is one of the body's largest continuous energy costs, which is why the metabolic state of a cell and its electrical state are never independent. And the scale is easy to underestimate. 70 millivolts across a membrane 7 nanometers thick is a field of roughly 10 million volts per meter, held quietly in every cell you have. Every tissue holds its own characteristic voltage. Red cells near -10. Keratinocytes -20 to -50. Hepatocytes -30 to -40. Beta cells and kidney tubular cells around -60 to -70. Neurons -70 to -80. Cardiac and skeletal muscle -85 to -95. A spectrum, not a single number. In excitable tissue, that resting state can be discharged. A stimulus depolarizes the membrane. At about -55 millivolts, threshold voltage-gated sodium channels open, and the interior swings positive within about a millisecond. Potassium channels then restore the resting state, and the impulse regenerates itself along the fiber. And once an action potential fires, its size is fixed. The information is carried in the rate of firing and in the rhythm of whole populations of cells, delta, theta, alpha, beta, gamma, which is precisely why frequency, and not amplitude alone, is a therapeutic variable. Cells are also wired to one another. Gap junctions let ions pass directly between neighbors, so coupled tissue behaves as a single electrical community. It is how a rhythm generated by a few thousand pacemaker cells commands a billion-cell heart, roughly 100,000 times a day. That rhythm is carried by the funny current, and ivabradine, a prescribed medicine, works by inhibiting it. Pharmacology, whose molecular target is a current. There are also diseases whose lesion is electrical. Inherited ion channel defects cause the long QT syndromes, several epilepsies, the myotonias, cystic fibrosis, and neonatal diabetes. And in the pancreatic beta cell, glucose depolarizes the membrane, calcium enters, and insulin is released. Stimulus secretion coupling, the glandular arm of bioelectricity. Vessel diameter is set the same way. A few millivolts of depolarization constrict an artery, a few of hyperpolarization dilate it. And force itself is converted into current by piezo channels. The 2021 Nobel Prize, including the baroreceptor reflex that reads your blood pressure on every beat. Even time is kept electrically. Neurons of the suprachiasmatic nucleus express the hour of the day in millivolts, and the nervous system regulates inflammation directly. Tracy described a vagal reflex that governs cytokine release. Stimulating that nerve inhibited TNF and reduced disease activity in rheumatoid arthritis, and in a 242-patient randomized trial published in 2025, improved it against sham, a therapy now approved. Electrical signal in, immune chemistry out. The clinical thread through all of this is that membrane voltage changes with disease. Depolarized states favor proliferation. Bioelectric dysregulation is described in cancer biology. Senescent cells sit depolarized. That is a serious research field, and it is still association and mechanism, not a demonstration that restoring a voltage from outside the body treats the disease. A medicine, meanwhile, only works if it reaches its target. Substances cross the membrane by passive diffusion, facilitated transport, active transport, ion channels, receptors, and specialized transporter proteins. Several of those are voltage-sensitive or gradient-dependent, so the electrical state of a membrane is part of the environment in which a drug acts. That is not the same as a mechanism for pushing medicines into cells, and it should never be described as one. Tissue itself has electrical properties. Cerebrospinal fluid and blood conduct well. Fat, bone, and dry skin conduct poorly, and impedance falls sharply as frequency rises. The same applied signal, therefore, produces very different current densities in different tissue. Intact skin is, quite literally, a battery. Epithelium pumps sodium and holds a transepithelial potential of 10 to 60 millivolts. Break it, and current flows out through the wound, setting up a lateral electric field of 40 to 200 millivolts per millimeter. Keratinocytes, fibroblasts, and immune cells migrate along that field, with a threshold near 25 millivolts per millimeter. Chronic wounds show a collapsed field. How much current and for how long decides everything. Nerve responds to pulses of tens of microseconds, muscle to hundreds, cardiac tissue to milliseconds. Perception begins near half a milliamp. Ventricular fibrillation risk begins near 50. Microcurrent therapy operates far below that, in microamps. The literature on low-intensity stimulation is real, but heterogeneous. Microcurrent and TENS-type stimulation have been studied for chronic wound healing, pain modulation, local perfusion, and neuromuscular reeducation. Cheng's 1982 experiment reported increased ATP at microampere currents, with the effect lost at higher ones, a laboratory finding, not a clinical one. And the honest record includes what did not work. Cochrane reviews found the evidence did not support TENS over placebo in chronic low back pain. A large international trial found transcranial direct current stimulation no better than sham, and another found it did not match an antidepressant. Interferential current helped as an adjunct, but not on its own. Samples are often small, and evidence for a modality class is not evidence for a specific device. Altered membrane potential in disease is an observation, not a diagnosis. Which brings the discussion to parameters. Voltage, the potential applied. Current, the charge that actually flows here in the microcurrent range. And frequency, the rate at which the signal pulses. And the parameters are the therapy. Frequency, waveform, amplitude, electrode configuration, tissue properties, exposure duration, and clinical context each change the result. Which is why this is a therapeutic signaling concept, titrated for an indication, not a frequency that commands an organ.
Is eMedica, a non-invasive platform built on controlled voltage, current, and frequency.
According to the manufacturer's proposed mechanism, controlled combinations of those three parameters are intended to support normal cellular bioelectrical activity, and through it, the process's repair depends on. That is design intent, not a demonstrated clinical effect. Precision matters more than enthusiasm here. Insulin resistance, for example, is impaired cellular responsiveness to insulin, a state involving receptor signaling, ectopic lipid, inflammation, and mitochondrial function. It is not simply fat sitting around a cell, and no current burns it away. The defensible proposition is narrower, that electrical stimulation may be investigated for effects on cellular processes relevant to insulin sensitivity. And electrical therapy is not an alternative to medicine. It is already part of it. Cardiac pacemakers and implantable defibrillators, cochlear implants, deep brain stimulation for Parkinson's disease, spinal cord stimulation, vagus nerve stimulation, functional electrical stimulation, bone growth stimulators, and electrical stimulation in chronic wound care. It goes further than symptom control. Approved electrical devices restore the work of failing organs. Implanted phrenic nerve stimulation restored breathing in central sleep apnea, 51% of treated patients against 11% of controls. A retinal prosthesis restored a form of sight. Sacral nerve stimulation restored continence in 83%. Catheter ablation abolished the arrhythmia substrate in 99% of cases. Bone makes the point most cleanly because bone is piezoelectric. Loaded collagen generates charge, and that charge steers bone-forming cells. Pulsed electromagnetic fields raised spinal fusion from 65 to 92% in a randomized trial, and a meta-analysis of 22 trials in nearly 1,500 patients raised fracture union from 64 to 80%. The first bone growth stimulator was approved in 1979. The largest electrical trials in medicine are cardiac. Implantable defibrillators reduced all-cause mortality by 23% across 2,500 patients. In oncology, alternating tumor-treating fields extended median survival from 16 to nearly 21 months in a phase 3 trial. There is a nutritional edge to this, and it is chemistry again. The sodium-potassium pump runs on magnesium-bound ATP. Coenzyme Q10 helps maintain the mitochondrial membrane potential that produces that ATP. So repletion of the substrates an electrically active cell depends on belongs in the same picture, physician-directed, and not self-prescribed. So a complete model of care has more than one dimension. Chemical therapy addresses biochemical pathways. Electrical therapy addresses bioelectrical and electrophysiological processes. Physical rehabilitation restores function. Nutrition supplies the substrate all three depend on. No single dimension is the whole of healing, and none of them replaces another. eMedica's own record is deliberately modest. 4,651 counted patients across 37 institutions with no serious device-related adverse events reported. Read honestly, that describes real-world usage and tolerability. It justifies formal study. It does not, by itself, establish disease-specific efficacy. 3 tiers, never blended. The physiology is established science. The wider stimulation literature is published evidence with known limitations, and eMedica's specific parameter combinations are a manufacturer-proposed mechanism. It is not for use with implanted electronic devices, over an active malignancy, or on broken skin, and needs caution in pregnancy, epilepsy, thrombosis, and impaired sensation. Reported effects of surface stimulation are mild and local. It is regulated as a class a non-invasive medical device under India's Medical Devices Rules, 2017, licensed by FD Maharashtra, with electrical safety tested at a NABL-accredited laboratory. What would settle the remaining question is ordinary science. Sham-controlled randomized trials, indication by indication, with pre-specified clinical endpoints, published stimulation parameters, and registry data from real use. Until then, the honest statement is a mechanism, a safety profile, and an adjuvant role, offered alongside the treatment a physician has already prescribed.
eMedica is complementary. It is designed to support medical care, never to replace it. Chemical medicine, bioelectrical support, and physical rehabilitation together. A more comprehensive approach to patient care. Electrical stimulation is indication and parameter specific. Evidence for one modality should not be generalized to another. Always used under qualified healthcare supervision. eMedica. Science, balance, healing. Always used under qualified healthcare supervision. eMedica. Science, balance, healing.
Everything in this film comes from published literature, textbooks, and regulatory records, and none of it should be taken on our word. The studies behind each statement, the textbook references, and the evidence tier assigned to every claim are compiled in the eMedica Bioelectric Compendium, published in full on the eMedica page on LinkedIn. Read it, check the citations, and judge the science for yourself.